Good morning to all of you. Last time we have seen the bacterial aerobic respiration and we have seen the part of glycolysis and TCA cycle, that is the Krebs cycle or citric acid cycle, and how the higher energy molecules like NADH and FADH2 are being generated and they are now connected into electron transport chain. Now let us understand the electron transport chain. The electron transport chain, it consists of a sequence of carrier molecules that are capable of oxidation and reduction. The electrons are passed through the chain. Stepwise release of energy takes place, which is used to derive the chemoosmotic generation of ATP. So ATP generation takes place in this particular phase. The final oxidation is irreversible. In eukaryotic cell, the electron transport chain is contained in the inner membrane of mitochondria. In prokaryotic cell, it is found in the plasma membrane. So if you see here, from this glucose metabolism, CO2 is being generated, and once the electrons are being chained or they are being cyclized. See, in a cyclic manner, they have been due to oxidation reduction reactions. They are now being undergoing with the electron transport chain and generation of ATP molecule takes place. There are three classes of carrier molecules in the electron. One is the flavoproteins, contains flavin as coenzyme derived from riboflavin B2, capable of performing alternative oxidation and reduction reactions. Important flavin coenzyme, flavin mononucleotide, FMN it is called as. Cytochromes, proteins with iron containing group, that is heme group, capable of existing alternatively in the reduced form Fe2+ and oxidation Fe3+, ferric and ferrous. Cytochromes involved in the electron transport chain include cytochrome B, cytochrome C1, cytochrome C, cytochrome A, and cytochrome A3. Ubiquinones and coenzyme Q and small non-protein carriers are also there, by which the electrons are transported under oxidation reduction reactions. See here. FMN oxidation reduction takes place. Q protein, cytochrome B, cytochrome C1, cytochrome C, cytochrome A, cytochrome C, A3. Ultimately, with the two hydrogen molecules and oxygen molecules, the water molecule has been released. During this whole sort of phase, the oxidation reaction takes place and formation of ATP has been generated. The final acceptor is oxygen, from which the hydrogen or the water molecule has been generated. The chemoosmosis, the mechanism of ATP synthesis using the electron transport chain. The movement of material across membranes from areas of higher concentration to areas of lower concentration, this diffusion yields energy. Energy release is used to synthesis of ATP. So this is how the actually model works on top of it. See here. There is inside the cell the mitochondrial matrix, and ultimately from here the formation of ATP molecule takes place, and this is the bimembrane in which this whole sort of high energy compounds are present, wherein oxidation reduction reaction takes place. Final acceptor is the oxygen, water molecule has been released, and 3 ATP molecule has been generated. Now total yield. Oxidation of glucose to pyruvate 2 ATP, substrate level phosphorylation. 2. Production of 2 NADH, 6 ATP molecules. Preparatory phase, formation of acetyl coenzyme A, 2 NADH, 6 ATP molecules. Krebs cycles, 2 GTP molecules equivalent to ATP. Production of 6 NADH, 18 ATPs. Production of 2 FADH2, 4 ATP. And this is total 38 ATPs are being generated when a molecule of carbon undergoes glycolysis, undergoes TCA cycle, that is the Krebs cycle, and undergoes the electron transport chains to form 38 molecules of energy. So this is how the overall reaction takes place inside the aerobic respiration in case of prokaryotes. That is, from one molecule of glucose, 38 ATP molecules have been generated. Correct? So that's all for today, and we have completed the part regarding aerobic respiration in bacteria.